Hello guys, you are welcome to this lecture. In this lecture, we are going to be talking about the amino acid, the structures of the amino acid, the classifications, and the general reactions that the amino acids undergo. Now, when we talk about the amino acid, what do we mean? Now, we have different biological macromolecules. Let's take the word macromolecules. Macro means large molecules. And these large molecules, we have carbohydrates, we have the proteins, and we have lipids and nucleic acids. These are the macromolecules. Each of these macromolecules, as the name is, like macro means they are large molecules. They are comprised of smaller molecules joined together by different kinds of bond. For instance, all carbohydrates consist of monosaccharides. That means smaller molecules joined together to give larger molecules of carbohydrate, like the polymers, like starch, glycogen, etc. Now, the proteins are comprised of amino acids joined together. All proteins are comprised of amino acid joined together by a peptide bond. So let's take note of this. Now, the amino acids that are found in humans that are found in living organisms are about 300. There are 300 amino acids found in living organisms. How many are they? 300. There are about 300 amino acids found in non-living organisms. But out of these 300, only 20 are the essential, or only 20 join together in different proportion to form protein. So we can say that protein synthesis is brought about by only 20 out of the 300 amino acids that are found in living organisms. So that is all about the description of amino acid as monomers of proteins. So whenever two amino acids, or let's say when amino acid combine with another amino acid, let's say the chain continues like that, it forms what? Proteins. So when amino acid combines together in different proportion, they form what? Or in different sequence, they form proteins. So let's continue with our study. We now have the structural description of amino acids. What are the amino acids made up structurally? Now, the amino acids are made up of the carboxylic acid group, which is in most cases is usually in the ionized form, and then it contains the amine group and then hydrogen, and then we have another group under here which is called the RH group, or the side chain. Now, what is the usefulness of the side chain? Every amino acid differs from the other with the side chain. Every amino acid differs from the other with the side chain. That is to say that if an amino acid is given, from here up, which is this other group, would be the same, but the side chain will vary. For instance, if I give you another amino, let's say if I give you an amino acid, the simplest amino acid, glycine, is COO minus, then we have NH3 plus, and then we have H, and then H. This is an amino acid called glycine. And then we have another HCOO minus, and then we have H and what? NH3 plus. Okay, let's change it to CH3. This is another amino acid. The name of this amino acid is alanine. Are we getting it now? So these are two amino acids. Now structurally, if you look at these two amino acids, you discover that this amino acid is different from this by only this group here, which is the what? The side chain. That is the only distinguishing factor. Now the side chain here is hydrogen, while the side chain here is what? A methyl group, which is CH3. While from here upwards still remains the same. From here upward still remains the same. So all amino acid differs from the other with just the side chain. Glycine, which is an amino acid, this is an example of an amino acid, is different from alanine, which is an example of another amino acid only by what? Changing the side chain. This one has hydrogen on the side chain, and this has CH3 on the side chain. So that is the structural description of what the amino acid is all about. Now, most amino acids are regarded as alpha amino acids. Why do you think they are regarded as alpha amino acids? Now, the reason why they are alpha amino acid is because if you look at this structure very well, let me redraw the structure of an amino acid here. Let's say you have this COO negative, and then you have C, and then you have NH3 positive, and then you have what? Hydrogen, and then you have CH3. The reason why this is called an alpha amino acid is because both the carboxyl group and the amino group are attached to an alpha carbon. Now, when do we say a carbon is an alpha carbon? For instance, let's use a normal straight chain to analyze this. If you have a carboxylic acid, this is COOH, and then it is attached to other groups like methyl groups. Now, from our basic understanding of organic chemistry, we said that the group, this is the preceding functional group here. Preceding because the carboxylic group has the highest what? Priority. Are we getting it now? Preceding because the carboxylic group have what? The highest priority. You understand now? So because the carboxylic group have the highest priority, what we are going to do is every group that is directly attached to the group with the highest priority is regarded, or to the functional group is regarded as what? The alpha carbon. Are you getting it now? So in this case, this group here is the alpha, while this group here is what? Beta, and this group here is what? Gamma. Same thing is applicable here. Now, in these functional groups, there are two functional groups present in all amino acids. Maybe I failed to explain that. There are two basic functional groups present in all amino acids. They contain the carboxyl functional group and the what? The amino functional group. Are we getting it now? They consist of the carboxyl functional group and the amino functional group. Now, what we are going to do is to analyze the two functional groups, the carboxyl and then the amino. Which do you think we have the highest priority among these two functional groups? Is it the carboxyl functional group or the amino functional group? The carboxyl functional group have the highest priority. Hence, the carboxyl functional group will take the functional group numbering, or the parent name of this amino acid. That is why they are called amino acid. That is amino group attached to a carboxylic acid chain. Are we getting it now? So the carbon that is directly attached to the carboxylic functional group is what is called the alpha carbon. Are we getting it now? So this carbon here is called what? The alpha carbon. Why this carbon here is called what? Beta carbon. So whenever the carboxyl group and the amino group are attached to the alpha carbon, we call this what? Alpha amino acid. You get the point now. So whenever we have the carboxyl group and the amino group attached to the alpha carbon. It is called the what? Alpha amino acid. So that is why these amino acids are usually regarded as alpha amino acid, because they have the carboxyl and the amino group attached to an alpha carbon. So that is that about the structural description of all amino acids. So if you are asked, how do you describe structurally an amino acid? I believe that you should be able to give a brief description of what it is. All amino acids have the carboxyl functional group and they have the what? Amino functional group. And we say that when the carboxyl functional group and the amino functional group are attached to the same carbon, which is the alpha carbon, we say that that carboxylic or that amino acid is known as the alpha amino acid. And we also said that the distinguishing factor between one amino acid and the other is the what? The side chain. The next thing that we are going to be talking about now is the classification of amino acids. The classification of amino acids. Now, amino acid may be classified as aliphatic amino acid. Aliphatic amino acids. Now, when you say something is aliphatic, it means it is not what? Aromatic. So, aliphatic amino acid are those amino acids that are not what? Aromatic. Are we getting it now? Now, under the aliphatic amino acid, we have classification. Under the aliphatic, we have the monoamino, then what? Monocabazolic amino acids. Under aliphatic, we have the monoamino, monocabazolic amino acids. Now, what are the monoamino, monocabazolic amino acids? They are those amino acids that have one amino group and also how many carboxylic group? One carboxylic. That is why they are called monoamino and what? Monocarboxylic amino acid. So any amino acid that have one amino group, any amino acid that has one amino group and one carboxylic group is known as a monoamino, monocarboxylic amino acid. Now, under this classification of monoamino, monocarboxylic group, we also have what is called the simple amino acids. Under monoamino, monocarboxylic, we have what is called the simple amino acids. Now, under the monoamino, monocarboxylic acids, we have the simple amino acids. And there are how many simple amino acids? Two. Which is what? Glycine and alanine. So we are going to draw the structure of glycine and alanine. Glycine is COO minus. Are we getting it now? Yes. Then we have what? C. And this is NH3 positive. And then we have H and H. Now this is the structure of glycine. This is the structure of glycine. So under monoamino, monocarboxylic amino acid, we have the simple amino acid like glycine. Then we now have alanine also. Alanine is C, which we've drawn before. This is negative. And then you have NH3 positive and H and then CH3. And then this is structure is called what? Alanine. Now these two amino acids form the basis of most other amino acids, especially alanine. Alanine form the basis of almost all other amino acids. Is that clear? So we have under simple amino acid, there are just two of them, which is glycine and alanine. Now, in most cases, some people may be confused. Like in some textbooks, you see the arrangement of the carboxyl functional group and then the amino functional group is not always in this position. Now, any position it is what? Correct. For instance, you may see it drawn like this. They may want to draw it like this. This is hydrogen, and then C, and then here is what? NH3 positive, and then we have COO minus, and then you have H, all so that it won't look so weird, the structure. Let's put COO minus here. We have COO minus and what? NH3 plus and what? Arrow, which is the side chain. So whichever way it is, it's actually correct. Now, in some cases, they may not write it in the ionic form. You may see it as H, and then what do you have here? C. If it is not in the ionic form, it means we'll not be having negative ion here, right? It means it will also be in the protonated form, which is COOH, and then here is what? NH2, and then you have what? R. Now, the reason why I wrote H2N is because is to show that the nitrogen is the one that is directly bonded with the what? With the carbon. Are we getting it now? To show that the nitrogen is the one directly bonded with the carbon. That is why I put H2N. H2N is still the same thing as what? NH2, which is the amine functional group. So based on aliphatic amino acid, which is based on structure, we have the monoamino monocabazolic. Under monoamino monocabazolic, what do we have? Simple amino acids. And under the simple amino acid, we've drawn the structure of two of them, which is what? Glycine and alanine. Now, we are now going under this, still the same. So, classification based on also the monoamino monocabazolic, we have the branched chain. After the simple amino acid, we have the branched chain amino acid. Let me use the blue pen since we are using that to write. Branched, it doesn't really mean anything, but so that the board will be colorful. Branched chain amino acid. Now, under the branched chain amino acid, we have three. The three are valine, isoleucine, and what? Lysine. You may use the acronym VIL, where V means what? Valine. V means what? Valine. Then I is what? Isoleucine. And then finally, L means what? Lysine. So let's draw the structure of each of them. This is I, isoleucine, and L-leucine. Let's draw the structure of each of them. First of all, we know that the upper part does not change. So we have COO negative, isn't it? Then we have C, and here becomes what? NH3 positive. And here is what? What do we have here? H. And then what do we have here? CH2. Then what do we have here? CH2. What other thing again do we have? Now, in this case, it's usually written as CH3, and then here is what? CH3. And this is the structure of valine. So you should be able to memorize the structure of the first 20 or the 20 essential amino acid, or let's not just say the 20 essential, the 20 important amino acid responsible for protein synthesis. 20 amino acids. The 20 amino acids responsible for protein synthesis. So this is the structure of valine. This is the structure of valine. Structure of valine is branched. That is why it is classified as a branching amino acid. If you look at this carbon here, it is a branched CH3. This is the straight chain. Then you have another CH3 here at this corner, which is what? Branched. Hence, it is considered as a branching amino acid. Exactly. Now, the next one we are going to look at is isoleucine. Now, look at isoleucine structure. It's written as, we should go white. What are we having here? COO minus. So here becomes COO minus, with COO negative. C. Then what do we have here? NH3 positive, and then what do we have here? Hydrogen, and then what do we have here? CH3, then what? CH2, and what? CH3. Now the difference between valine and isoleucine, can be seen in this CH3 group. You see valine, the chain ended here. Are we getting it? While isoleucine have another methyl group attached to it. Are we getting it now? So that is valine. Now, if you look at it very well, it is also branched, like in the case of this one. So all these ones that have a chain that deviates from the original long chain structure are known as what? The branch chain amino acids. Now, this to say they have a side chain that is branched. Now, let us look at the last one. The last one is isoleucine. Now, the word iso means what? The same thing. So, isoleucine is the same thing as what? Leucine. Like isomer, the same, but different structural arrangement. So, in this case, what differs is where they branched. Are we getting it now? So, let us draw the structure of isoleucine. You see that they have the same molecular formula, the same everything, but the structure is only different at what? At the position of the branch something. So we have here COO minus, then what do we have here? C, then NH3 plus, and here is what? H, and then what do we have here? CH2, CH, then what do we have? CH3, and then CH3. Now, they are the same, but the only distinguishing factor here is while the CH3 is, if we want to number, we say this is number what? What will be this number? Number 1. Here is what? 2. Here is 3. Here is 4. Here is 5. We can use the alpha, beta, gamma, whatsoever to number. Same thing applicable here. Here is 1. Here is 2. Here is 3. Here is 4. Here is 5. Now you discover that the both of them, they have 5 chains or 5 straight chain, then a branch chain. Now the branch here is at position number what? 3. Are you getting it? Why the branch here is at position number what? 4. That is the only distinguishing factor. That is why this one you have isoleucine and leucine. So we can say leucine and isoleucine are the same, but only varies in the position of a methyl group. Is that understood? Now, after the branching amino acid, we have the... So, let's look at another classification of the amino acids. Another classification. All these ones, they are called the Hydrozi amino acids. Now, as the name implies, hydrozi means those ones that contain what? OH. Now, all hydrogen amino acids have their roots, or let's say the first member of each of these categories have their roots from analyne, but one of the hydrogen must have been what? Substituted. So if we take out alanine, what is the structure of alanine again? Alanine is COO negative. Then we have C, and then we have what? NH3 positive, and here is what? Hydrogen, and then we have what? CH2. Are you getting it? CH2 and what? OH. Now this amino acid is called serine. Now, if we should come here and draw the structure of alanine. Alanine is COO negative. This C, this NH3 positive, and then we have what? H, and then we have what? CH3. Abi? So when one of the hydrogen, if we remove one hydrogen from here, we can write it as, instead of CH3, we can put it as what? CH2 and what? H. So if this hydrogen here has been substituted with OH, that amino acid that is formed is what is called what? Serine, which is a hydroxy amino acid. Are we getting it? Hydroxy containing amino acid. Now the next one we are going to look is threonine. Threonine is almost the same thing with this. What is the structure of threonine?
COO minus.
This will be what? COO minus. Then what do we have here? C. And then we have what? NH3 positive. And then we have what? Hydrogen. And then we have CH2. Then CH2. And then finally what? OH. The differences between serine and threonine is this threonine is just one CH2 group added more to serine to give us threonine. So it is essential that you know how to draw these amino acid. So these are the two hydroxyl amino acids. So we've learned how many? How many of them? We've learned about 5, which is simple amino acid, glycine and alanine. We've learned about the branching amino acid, valine, isoleucine, aleucine. And then we've learned about the what? The hydroxyl amino acid, which is serine and what? Threonine. 7. So let us look at the sulfur containing amino acid. The sulfur containing amino acid. Sulfur containing amino acid. Let's look at the sulfur containing amino acids. Now sulfur is X, so any one of them that have this XH configuration is part of the sulfur containing amino acid. Is that clear? So let us draw them now. The first one that we are going to look at is what? Cysteine. There are 2 sulfur containing amino acids, cysteine and what? Methionine. Cysteine and methionine. So let us draw them together. What will be their structure? Can we go together? This is what? COO negative. Then what do we have? C. Then this is NH3. What do we have here? Positive. And then H. And then we have CH2 and what? SH. So you can see that is similar to serine. But the difference is that in place of this OH, we are going to put what? SH. That is the only difference between this and the other one. Okay, let us draw methionine. Methionine is COO negative. Then what do we have here? C, NH3 positive. Then what do we have here? H, and then CH2. And then we have another CH2, and then we have what? S and CH3. This is the structure of methionine. Are we getting it? So these are the 2 amino acids that are sulfur containing amino acids. So let us look at the next. The next we are going to look at are the amines. The amines containing amino acid. Amine containing. Now the question is, what is the structure of the amine? The amine, they usually have R, C, double bond, then what? NH2, which we can write in the condensed form as ROCO and what? NH2. So any amino acid that has this form is said to be the amine containing amino acid. And there are 2 of them, which is what? Asparagine. There are 2, which are asparagine. And then we have what? Glutamine. We have asparagine and then we have glutamine. Are we getting it now? So let us draw the structure of each of them. Now, asparagine still gets its structure from alanine. What is the structure of alanine the other time? COO negative. Then we have what? NH3 positive. And then we have H. And then we have CH3. So when one of these hydrogen has been substituted, it means we can have CH2 here. Then the group will be attached here. Are we getting it now? So let us come here and write the asparagine. Structure will be COO negative. Then we have what? C NH3 positive. Then we have what? Hydrogen. And then we have CH2. And then we have CONH2. So this is asparagine. you can see the amine functional group here. The amine functional group here. So that is why we call them the amine containing amino acid. So whenever you see any amino acid at all that has this CONH2, such amino acid is called the amine containing amino what? Acid. It's as easy as that. Are we getting it now? So let us talk about the next. The next one is glutamine. What is the structure of glutamine? Glutamine is almost the same thing as what? Asparagine, but the only difference is the addition of one CH2. So let us draw and then see what it looks like. This will be COO negative. Then what do we have here? C, and then what? NH3 positive, and then we have H, then we have CH2, then we have CH2, and then we have COOH. Sorry, CONH2. This is the amine functional group. So this is glutamine, and this is asparagine. So any amino acid at all that has the amine functional group, is said to be the amine containing amino acid. Is said to be what? Amine containing amino acid. Now the next one we are going to look at are the acids, or we've talked about monoamino, monocabazolic. So under monoamino, monocabazolic, we talked about the simple amino acid. Under this one also we talk about the branching amino acid. We talked about the hydroxyl amino acids. We talked about what? Sulfur containing amino acid. And then we have the amine containing amino acid. Now you discover something, that each of them that we've been talking about, they have only one function, or they have only one carboxylic acid group, and also only one what? Amino group. So each of these ones that have only one carboxylic and only one amino group are classified as what? Monoamino, then mono what? Carboxylic, or monocarboxylic monoamino acid. Are we getting it now? So that is that for this session. Now let us look at the next.
Monoamino monotype.
Now we have the next, which is
monoamino dicarboxylic.
Monoamino
Dicarboxylic.
Then we have what? Dicarboxylic amino acid. Monoamino dicarboxylic amino acid. Now, as the name implies, when we say dicarboxylic, what does it mean? It means if we have how many carboxylic group? Two carboxylic group. It means if you have how many carboxylic group? Two carboxylic group. Are we getting it? All these are actually said to be acidic amino what? Acids, because they have two carboxylic group. Are we getting it now? So this one has a carboxylic group in its side chain. Aside from the normal carboxylic group in the what? Amino acid. Are we getting it now? An example of them, we have two examples. The two examples are what?
Glutamic and aspartic.
We have a glutamic and what? Aspartic acid, or we can take it as aspartic and glutamic what? Acid, so that we'll not be making mistake. Aspartic, and then we have what? Glutamic amino acid. Now, under aspartic is gotten from asparagine. When this group here is changed to a carboxylic acid group, asparagine becomes what? Aspartic. So if we draw the structure of asparagine and change this group here just to COOH, it becomes what? Aspartic. So we can say that aspartic is gotten from what? Asparagine. So this becomes COO negative. Then what we have here? NH3 positive. Then what we have here? H, and then CH2 COOH. Are we getting it now? So this one is what? Aspartic acid, gotten from asparagine. Are you getting it now? Same thing applicable here. Now, glutamic acid will be gotten from what? Glutamine. To be gotten from glutamine. Just change this CONH2 to COOH. So this gives us, we have COO negative, and then we have what? C. Then what do we have here? NH3 positive, and then we have what? H. Then CH2, COOH. This is glutamic acid. So when we talk about monoamino dicarboxylic amino acid, we have two categories. Are we getting it now? The two categories are, two examples are what? Aspartic acid and what? Glutamic acid. We have the what? Aspartic acid and glutamic acid. So let us go to the next classification. We've talked about monoamino dicarboxylic. Now, what do you think can be the other form of monoamino and dicarboxylic? It will be what? Diamino or dibasic amino acid. Dibasic. That means they have two amino group, or you can take them as dibasic amino acid. They are classified as what? Dibasic amino acid. That means they have how many basic group? Two basic group. Dibasic amino acid. They have two basic group. And in chemistry, organic chemistry specifically, when you talk about base in organic chemistry, the group of compound that is often referred to are the what? Amine. Are we hearing it? The amine functional group. When you talk about the basic group in organic chemistry, you're talking about the amine. So when we talk about dibasic, it means they contain they contain more than two rather, since it's di specific. So it contains two amino groups. Are we hearing it now? Two amino groups. Please take note of the word group. That is why I actually use the word group, because there may be more than two amino the two amino molecules there. There may be more than two, but one of them may be considered as a group and the other as another group. Now let us go. Under this one we have two examples, which are what? Arginine and what? Lysine. Now I usually give a way to write this. Now for this one, all you need to write, they are very long. So first of all, we have COO negative, C, then NH3 positive, then what do we have here? H, then what do we have here? CH2. Now, after writing the main group, the side chain, before you add anything, please write 3 CH2s so that you remember. So this is what? CH2. Then this is what? CH2. And then here you have what? N. Are you getting it? N, H. Then here you have what? C. Double bond what? NH2 positive. And then what do we have here? NH2. Now this is the structure of arginine. Arginine. Now this is the structure of what? Arginine. Yes. You're asking me if I recorded it. This is the structure of what? Arginine. Now the arginine contains a special group that is called the guanidinone functional group. That is called what? Guanidinone. This functional group here is what is called the guanidinone functional group. So in case you are asked to draw an amino acid that has the guanidinone functional group, you should be able to draw it. This functional group here is what is called guanidinone functional group. So you have or that group there is what is called the guanidinone. They are often referred to as what? Special groups. Special groups. So this is the guanidinone group, which is often referred to as special group, which is NH, and then you have C double bond N, H2 positive, and then you have what? NH2. This is arginine. Now, if you look at the structure of arginine very well, let us look at something. That was why I was telling you that it's not usually the amine, but the groups. Now, this NH3 plus is classified as one amine group. Are we getting it now? And this arginine or this guanidinone group is classified as another word? Amine group. So you see that in this case, that amino acid has two amine group. Hence, it is said to be dibasic amino acid. Is that clear? Now, the next one we are going to be taking is lysine. What is the structure of lysine?
C-O-O minus.
What is the structure of lysine? You have it as what?
C-O-O
This
negative.
COO negative. Then what do we have here? C. And then what do you have here? NH2, NH3 positive, NH3 positive. Then what do we have here? H. And then CH2, CH2. We said we must write 3 CH2. And then what do we have here?
You have NH2.
NH2. Okay. This is lysine. This is structure of lysine. Are we getting it now? This structure. Now, if you look at the structure of lysine, you see that it contains, this is an amine group, does not contain special group, and this is another amine group. So lysine consists of 2 amine group. So that is for dibasic amino acid. So on aliphatic amino acid, we are done with them. Under aliphatic, we are done with them. So under aliphatic, we say we have monoamino monocabazolic, which we talk about the simple, the branched, the hydroxylamine, the sulfur containing, the amine containing, and then we went to monoamino dicarboxylic amino acid. We talked about 2. We talked about the dibasic amino acid. We talked about 2. Now in this one, we are going to be talking about the aromatic amino acid. Aromatic amino acid. Now, when we talk about aromatic, most times we usually sense benzene structure first before anything. So under the aromatic amino acid, we have 2 to be classified under there. Now, the 2 of them that will be classified under the aromatic, what will be their name? Phenylalanine and what? Tyrosine. There are only one difference between them. Phenylalanine and tyrosine. So we have these 2 amino acids as the aromatic amino acid, phenylalanine and tyrosine. Phenylalanine and tyrosine. So we have phenylalanine and tyrosine. Now let us draw the structure. We have the benzene. As the name implies, phenyl means you have the phenyl ring. So let us draw the structure of benzene. Now, if this is the structure of benzene, then we have the alanine group attached with H2. Then we have what? COO minus. And then what do we have here?
We have H.
Are we supposed to have this? No. Supposed to come here first. C.
C-O-O.
Then COO what? Negative. And then we have let's put the H3N positive, and then we have what? CH3. This is structure of phenylalanine. Are we getting it now? Now this is phenyl ring. From here like this is a phenyl ring. And then if you look at this structure very well, this other structure is what? Alanine. So when a phenyl ring attached to the structure of alanine, it gives us what? Phenylalanine. It's aromatic amino acid because of the presence of the benzene ring. Are we getting it now? So that is for phenylalanine. Now the next one is tyrosine. Now the only difference between phenylalanine and tyrosine is the addition of what? OH. In most cases, tyrosine is classified also as hydroxyl amino acids. Are we getting it now? So we have this as, we have the structure. My something is looking very beautiful. So we have COO negative, and then we have what? NH3 plus, and then CH3. Then we have what? The OH. So this is tyrosine. The The difference between tyrosine and phenylalanine is this OH here. Are we getting it now? So whenever you get to phenylalanine, you should be able to draw the structure of what? Tyrosine. Is that clear?
Yep.
Should be able to draw the structure of tyrosine. So those are the aromatic amino acid. Two of them. Aromatic, we have the phenylalanine, and then we have what? Tyrosine. We have the phenylalanine, and then we have what? Tyrosine. Now let's all look at the next one. The next one is heterocyclic amino acid. Now the first question you need to ask yourself here is, what are heterocyclic compounds? Now heterocyclic compounds are compounds that have what? Heteroatom in the carbon chain. That have what? Heteroatoms in the carbon chain. When you say it has heteroatom in the carbon chain, it means it has other atoms like oxygen, sulfur, nitrogen, etc. on the main carbon chain. For instance, if you have a compound like this, normally this is supposed to be something like carbon, carbon, carbon. This is what? Cyclo 1, 2, 3, 4, 5. Cyclopentane, right?
Yep.
Then if you put this becomes cyclopentadiene, 2 in group. Now, in a situation where instead of, you know that at every point here there is a carbon. True or false? True. At every point here there is a what? A carbon. Now, instead of having a carbon there, how about we have, instead of having a carbon here, how about if we have something like oxygen here? How about if we have something like oxygen here? Now, this oxygen here becomes what? A heteroatom. It becomes what? Heteroatom. Are you getting the point now? So whenever you have other atoms aside from carbon in the chain, such is known as heterocyclic compound. And the two, we have about two heterocyclic compounds. We have or heterocyclic amino acid. We have tryptophan and what? Histidine. We have tryptophan and, so let's look at the structure of the two heterocyclic amino acid, tryptophan and histidine. Now, these two amino acids, they contain special group like that of the guanidinone functional group. They contain functional groups or special groups like that of the guanidinone. So let's write them down. We have tryptophan, and we have what? Histidine. Now, the histidine or tryptophan has what is called the Indo-functional group as a special group. Indo-special group. Are we getting it now? That's what? Indo. Now, what is Indo? Indo is drawn like this. You have your normal benzene ring, which is drawn like this. Pardon my drawing. And then you have another group which comes this way, then N, and then you have something like this. Now, this is the Indo-functional group. Then when you now have Indo plus alanine again, Indo plus alanine, this is H2, then this is C, this is what? H3N plus and what? COO negative, and then you have CH3. So whenever you have the Indo ring attached to alanine, it gives you what? Tryptophan. This is the Indo ring. It's a special group here. Are we getting it now? So whenever you are asked, what is the special group that is present in tryptophan? What will be your answer? It has the Indo special group. Now the next one has histidine, has the imidazole. Imidazole. Now when you have the word diazole or dazole, it's telling you that in the compound there are how many nitrogen? 2 nitrogens. Are we getting it? There are 2 nitrogens. So let us draw the structure of the imidazole group. Imidazole group is like this. Then you have our what? Nitrogen, H, and then you have what? H, and then you have something like this. This is the what? Imidazole functional group. Are we getting it now? And then you have what will be here? Alanine. This is structure of alanine again. C. Here will be H3N plus, and then COO minus, and then what do you have here? CH3. So this is the histidine. So at this point in time now, we've been able to draw the first 20 amino acids. Are we getting it now? The first 20 amino acids. These ones, they are heterocyclic. Now, why are they heterocyclic? As I've earlier said, I said heterocyclic are those ones that contain other heteroatoms like oxygen, sulfur, and nitrogen in the what? Carbon chain. So this is the carbon chain, supposed to contain carbon, but you now have this group here. Are you getting it now? So all these ones, they are what? Heterocyclic amino acids. So that is that for this session. Let us look at the amino acid. Amino acids. Now, as name implied, there is only one amino acid, and that amino acid is what? Proline. The amino acid is proline. And the structure of proline contains the pyrrolidine functional group. What is pyrrolidine? Pyrrolidine is the hydrogenated form or the saturated form of pyro. This is the structure of pyro. Is heterocyclic also. This is structure of what? Pyro. So whenever this one is hydrogenated, or if it is saturated, that means there are no double bond, it forms something like this. You have N, and then you have what? H. This is what is called the pyrrolidine functional group. Are we getting it now? And proline has the pyrrolidine functional group. Are we getting that now? Now, what will be the structure of proline? Proline, in most cases, is written as what? This, and then you have this group here. Sorry for the whites. You're supposed to use white to draw. Now, the structure of proline can be drawn in this way. First of all, by writing the amino acid, which is NCOOH. Then we have CH, and then we have H3C, or H2C rather, in order not to exceed the valency. And then it continues like that, H2C, and then this one also, you have CH2. Then here you have NH, and then you have CH2. So we can see that amino acid is also what? Heterocyclic. It's also heterocyclic anyway. Now, so this is the structure of proline. It's an amino acid. So you may be asked to draw the structure of an amino acid. What you are expected to give is the structure of what? Proline. The structure of what? Proline. Proline is an amino acid. So that is that for the structure of the first 20 amino acid. That is that for the structure of the first 20 amino acid. Now, let's look at amino acid based on other classification. We have the derived amino acids. Now, under derived, we have the ones that are derived from proteins. Are we getting it now? So we can say they are the derived amino acid from proteins. Let's take them like that. We have the derived amino acids from proteins. Under this, we have two. We have the Hydrozi, Lysine, and then we have what? Hydrozi, Lysine, and we have what? Hydrozi proline. Under this classification, we have the Hydrozi Lysine and the Hydrozi proline. Now, this derived amino acid is after protein synthesis. those amino acids are modified to give the derived form. For instance, after the synthesis or after the metabolism of protein, lysine is modified to give the Hydrozi lysine, and also proline is modified to give Hydrozi proline. So we said under the derived amino acids, we have derived amino acid from proteins, and we talked about example of those are Hydrozi lysine and Hydrozi proline. Now let's look at derived amino acid not found in protein. Derived amino acid not seen in proteins. Derived amino acids not seen in proteins. Now all these ones, they are seen in free cells. They are seen in free cells, not seen in protein. They are seen in free cells, like not seen in proteins. They are seen in free cells. Example is citrulline. We have ornithine. We have citrulline, ornithine, and homocysteine. All these ones, they are derived amino acids that are not seen in proteins, but they can be seen in the free cells. So all these are derived. They are seen in free cells. That's for that classification. Then we have the non-alpha amino acid under derived. We have non-alpha amino acid. The non-alpha amino acid. Now, under the non-alpha amino acid, we have example. We have the gamma amino butyric acid called GABA. Gamma amino butyric acid. Now, gamma amino butyric acid is gotten from the decarboxylation, that means removal of the carboxylic group of gamma amino butyric acid is gotten from the decarboxylation, removal of the carboxylic group of glutamine, of glutamic acid rather. When glutamic acid is decarboxylated, what is formed is what? Gamma amino what? Butyric acid. Butyric acid. So please, let's take note of all that. Are we getting it now? So that is for amino acid, or based on structure, we have the derived amino acid. So classification of amino acid that are derived. We say we have the ones that are derived from proteins and the ones that are derived but are not seen in proteins. And then we have the non-alpha amino acid that is gotten from gamma amino, that is gotten from what? Glutamic acid. Glutamic acid. When glutamic acid is decarboxylated, it forms a gamma amino butyric acid. Are we getting it now? So let's go to the next. Now, the special groups in amino acid, we've listed them before. We talked about the what? Guanidinone group. Where is the guanidinone group found? It's found in what? Arginine. Arginine. Are we getting it? Guanidinone group is found in arginine. What other group again? Indo group. Found in what? Tryptophan. Indo group found in tryptophan. Imidazole
found in histidine.
Imidazole group found in histidine. And then what do we have again? We have the imidazole found in histidine, indo found in tryptophan, guanidinone found in arginine, and then finally, I think we also have the benzene ring. Benzene ring is found in 2 amino acid, which is what? Tyrosine and phenylalanine. Tyrosine and phenylalanine. Phenylalanine. And then we have the pyrolysin group. Pyrolysin group. Where is the pyrolysin group? In proline. So these are the special groups found in all amino acids. Special group found in all amino acids. So that is that. You should be able to identify them. Now the next thing we are going to look at is based on side chains. There are some amino acids that have non-polar side chains. They have non-polar side chain. Let's look at examples of such amino acid with non-polar. That is to say the side chains are non-polar. Now, if you take a look at alanine, is non-polar because it contains only carbon and what? Hydrogen. When you see something is polar, it means it has something like a more electronegative atom like oxygen attached to something. So these ones are what? Polar. Are we getting it? If you have like OH attached or a more electronegative atom attached to any other thing, then such amino acid is polar. But anyone that does not have anything like that, that is, if it contains only carbon and hydrogen in its side chain, such amino acids are said to be non-polar. An example, we have alanine, we have glycine, we have what? Valine, we have isoleucine and what? Lysine. Are you getting it? So all these ones, they are non-polar. Even what is it called? Methionine is also considered as non-polar. So let us give examples of non-polar amino acid that we know. Those ones that contain ring, that have only carbon and hydrogen, and others. So under this one we have alanine. What other example again? We have valine. We have what? Isoleucine. And then we have what? Lysine. Even methionine is considered as what? Non-polar. etc. All these are non-polar amino acid. So we have the ones that are having uncharged or non-ionic polar side. Now, before we talk about the other category, when we say something is polar, it means that the something is what? Hydrophilic. And when we say something is hydrophilic, what does it mean? It means it is soluble in what? In water. Are we getting it now? Hydro means water. Philic means what? Love. Are we getting it now? So hydrophilic means what? Water loving. Soluble in water. So all polar amino acids are what? Soluble in water. An example is alanine, valine, isoleucine, lysine, methionine, histidine, etc. All those ones, they are what? Amino acid having non-polar side chain. They are hydrophobic. These groups are hydrophobic. When you say something is hydrophobic, it means it does not what? Like water. Does not like water. And then it is what? Insoluble in water. So all non-polar amino acids are hydrophobic. They don't dissolve in water. They don't like water, and they are what? Insoluble in water. An example is everything that is listed here. Then we come to amino acid having uncharged or non-ionic polar side chain. Now, in this case, although the side chain is polar, but it is what? Uncharged or non-ionic. Non-ionic. And you can see them from the structures. Like, if we go back to the initial parts, we have these ones like cysteine. It is polar because we have sulfur attached to what? Hydrogen. Sulfur is more electronegative than hydrogen. Also, we have serine. You have oxygen attached to hydrogen. This is polar. Also, threonine. That is polar. Also, glycine is considered as that. Then tyrosine, glutamine, and asparagine. All these ones, they have polar side chain. Carbon bonded to oxygen, nitrogen. This one is polar. This group is polar. Then, so all those ones, they are examples of polar amino acid, but they are uncharged. That is to say, they don't have any charge in them. So here you have example. They gave glycine as an example here. So take note, glycine is not in the first category. Glycine is an example here. Then we have cysteine, which contain what? Sulfur and hydrogen. We have another category, threonine. Then we have serine. Then we have asparagine, glutamine, tyrosine that contains OH group. All these ones are polar, but they are non-ionic. That is, they don't have any charge. Non-ionic. If you look at their structure, they contain, like cysteine contains SH. Threonine contains what? OH. Also, serine contains OH. Asparagine contains CONH2. Glutamine, the same thing, COO or CONH2. Tyrosine contains OH. All these are what? Polar, but they are non-ionic. Are we getting this now? So let's go to the next. The next are amino acid having charge or ionic polar side chain. All these ones, they are the two categories, which are the diacid or dicarboxylic, and then we have the dibasic. Examples are, under these two categories, we have aspartic acid and what? Glutamic acid. Why? They contain two acidic group, which is C contains two acidic group, which is COOH. This COH can hydrolyze to give us what? COO negative. Same thing applicable here. This contains NH3, which or NH2, which when protonated will give us NH3 plus. Are we getting this now? So that is that for that quest. Now the next one, let us attempt to classify amino acid based on the mechanism. Now we said that proteins, when proteins are broken down, what are we form? We form amino acid. So whenever proteins are broken down, we form amino acid. How about when amino acid combine together? What do we form? We form proteins. Now, based on mechanism, we are talking about whenever these proteins are broken down, what mechanism does the amino acid, or what is formed from the amino acid that combines to form that protein? What is formed from the amino acid that combine to form that protein? So based on the mechanism, if they are broken down, what pathway do they follow? It is either they are ketogenic, or they are glucogenic, or they are both ketogenic and what? Glucogenic. So in this session, we are going to look at the ones that are purely ketogenic amino acid. Now, purely ketogenic amino acid are those ones that when they are broken down, they form ketone body. They form what? Ketone bodies. An example of these ketone bodies, we have acetone. What is the formula of acetone? CH3, C double bond O. There we have what? CH3. That is to say, when these amino acids are broken down, they form what? Acetone. or they can form keto bodies. An example of that keto body is what? Acetone. You can see from the C double bond O, this is ketone functional group. It can form acetoacetate. It also has the C double bond O functional group. At the same time, it can form hydroxy what? Hydroxy butyrate. All these are ketones. So all those amino acids that when they are broken down, they form ketone bodies. They are known as what? Ketogenic amino acid. They are known as what? Ketogenic amino acid. Now, more details about the metabolic pathway will be explained in details in our later chapter when we are talking about the macromolecule metabolism, like the carbohydrate metabolism, lipid metabolism, and protein metabolism. But for this case, let us just know that those ones that when they are broken down, they form ketone bodies are said to be what? Ketogenic. And the three ketone bodies that are formed are acetone, acetoacetate, and hydroxy butyrate. So what are those amino acids that when they degrade, they form these ketone bodies? What are they? Now, I like to use a mnemonic to actually remember them. Now, after K is ketogenic. Just use this to remember them. After K in the alphabet, what do we have? We have L. So the two amino acids that start with L are ketogenic. An example is lysine and leucine. These two amino acids are purely ketogenic amino acid. So based on metabolism or based on mechanism, they are classified as ketogenic, glucogenic, or a combination of both ketogenic and what? And glucogenic. A combination of what? Ketogenic and glucogenic. So let's continue. The next one is ketogenic and glucogenic. All these ones, they form ketone bodies, which are when broken down, and they also form what? Glucose. The ones that form glucose are said to be glucogenic. That is, if the amino acids are broken down, what do they form? They form glucose to release energy. That maybe in terms of when you are very, very hungry and you're not taking a lot of carbohydrate, these amino acids are converted to glucose in the body to supply energy. All those ones, they follow the glucogenic pathway. So it can either be ketogenic or glucogenic, or a combination of both ketogenic or the glucogenic pathway. Are you getting it now? So examples of the ones that fall into this category, I like to use the mnemonic, which is TTIP, where one of the T, let me write them down, TTIP. There are 4 of them. We have the first one, tryptophan. The second one, tyrosine. And then we have isoleucine. And then we have phenylalanine. All these are both ketogenic and glucogenic. TTIP, in case you forgot. TTIP. So whenever you just remember TTIP, you should be able to remember the first. Now, the first one we said ketogenic is LL. After K, you get to L. Leucine and lysine. Then both ketogenic and glucogenic. Note, I didn't go to glucogenic now. I went from ketogenic to both ketogenic and glucogenic. Is the fact clear? We went from ketogenic to both ketogenic and glucogenic so that you will not muddle up things later. So we have TTIP, where one of the T means tryptophan, the other means tyrosine, other means what? I means isoleucine, and P means phenylalanine. Now, the next category that we are going to be looking at is purely glucogenic. So all those that are converted to glucose only. Now, if you could look at what we did here, we had here how many of the amino acid? 4 plus Now, initially, how many did we classify? Lysine and leucine. That is how many? 2. 4 plus 2 is what? 6. And we had about 20 amino acids minus 6 that we've already classified, so we have 14. So it means that the 14 remaining amino acids are what? Are glucogenic, purely glucogenic. The 14 remaining amino acids are purely glucogenic. Now, under this, we have valine, alanine, leucine. Okay, we've talked about leucine, valine, alanine. We have threonine. We have serine, cysteine, etc. The remaining 14 amino acids, apart from tryptophan, tyrosine, isoleucine, phenylalanine, and lysine and leucines, are all purely glucogenic amino acid. So if you are asked to classify amino acid based on the metabolic pathway, we have the purely ketogenic, glucogenic, and ketogenic, and then we have the purely glucogenic amino acid. Is that clear? So let's go to another classification. Now we say we have based on the nutritional requirements. Based on the nutritional requirements. Now, based on nutritional requirements, we have about 3 classifications of the amino acid. 3 classifications. But here I gave 4, but it's supposed to be 3 classifications. Okay. Now the 3 classifications of the amino acids are: we have the essential or indispensable amino acid. Now, when do we say an amino acid is essential? Now, an essential amino acid are those that cannot be synthesized. Those that cannot be synthesized by the body, and hence should be taken in our diet, are said to be essential amino acid in our diet, or in diet, are said to be essential amino acid. So any of those amino acids that our body cannot synthesize, but they are needed by our body for metabolism and growth, those amino acids are said to be essential or indispensable amino acid. An example is, or let me explain again, those that our body cannot synthesize, but we need them in our body for the nutritional requirement, for metabolism and growth, for cellular activities. Those amino acids that our body cannot be synthesized but can be taken in are said to be what? Essential or indispensable. So whenever you eat food, one of such amino acid is being what? Added to our body. Are we getting it now? An example is, we have a short mnemonic or a memory aid to actually help in determining the ones that are essential and the ones that are not essential. We have any help in learning these little molecules proves truly valuable. So using this memory aid, let us name all the 10 essential. We have any. Now, any means what? Any means arginine. Okay? Any means arginine. That is the amino acid there. Now, help means what? Histidine. Then we have in. In means isoleucine. Then we have, after isoleucine, we have leucine for learning. Leucine. What is the problem of this stuff? We have leucine. After leucine, we have dis. Now, dis, we have threonine. From TH, we have threonine. Now, after threonine, we have lithu. Under lithu, Sorry about that. Under little, we have leucine. And then we have molecule. Molecules, we have methionine. Proofs, we have the phenylalanine. Truly. Now we have T's, about 3 of them in the amino acid. Have we named anyone? Yes, threonine. We have tryptophan. Truly. We have tryptophan. Valuable. We have valine. So these are the 20 essential amino acids based on the nutritional requirement. Any little help in learning these little molecules prove truly valuable. A is arginine, H is histidine, N is isoleucine, leucine, then this is threonine, little leucine. We've written leucine before, so any help in learning this little. The other little will be lysine. I think so. Be lysine, because we've written that before. So here we have lysine. So these are the 10 essential amino acids, those that the body cannot synthesize. Hence, we need to take them into our body for metabolism and growth. Now, let us look at the next classification: partial or semi-essential. Now, these amino acids are semi-indispensable. Are we getting it? Now, they may be required for children during their early stage of development, but as time progresses, they may not be required by the body again. Are we getting it now? So such amino acids are known to be semi-essential or partially essential. They may be essential at the early age of development of infants, but as they become adults, they may not be essential. So such amino acids are said to be partially essential or semi-essential. Now, the next is non-essential or dispensable. Now, these are synthesized by the body, hence you don't need to take them in as food. They are synthesized by the body. Now, we've written the 10 essential. So in order to get the non-essential, they are the other 10. The other 10 are what? Non-essential. So all you just need to do now is, we are now going to use the idea of set. If you have U as the universal set, and the universal set contains 20 amino acid, which is total. Now, the essential are what? 10 amino acid. What do you think will be the complement, which is E complement? E complement, which is essential, will be what? U minus E, and that will give us what? The non-essential. We are bringing in the idea of mathematics here. So what do we do now to get non-essential? The 20 amino acid minus the 10 essential will give us what? 10 non-essential. So having known the 10 essential, the remaining 20 amino acid are said to be non-what? Essential. Having known the 10 essential, the remaining 10 amino acids are said to be non-what? Essential. An example, we have proline. We have histidine. Did I write histidine for, okay, I wrote histidine for ketogenic, for help. Did I write histidine? Okay, histidine is there. We have proline. We have tyrosine. Then what other one again do we have under the non-essential? We have the asparagine, glycine, glutamic acid, aspartic acid, cysteine, All those are non-essential. Aspartic, okay, because the body can synthesize them. All these are what? They are non-essential because the body can what? Synthesize them. So the remaining 10, if you know the 10 essential by using the mnemonics or the memory aid, any help in learning this little molecule proves really valuable. All you just need to do is subtract them and you get the non-essential. Now the next one is the conditionally essential amino acid. Now let us say you have a full-grown human, okay? This is a full-grown human. Now something may happen, and then those amino acids that are non-essential becomes what? Essential. Becomes essential. Those that are non-essential, they becomes what? Essential. Now, why would you think that an amino acid that was non-essential? Now, non-essential means what? It means that they are synthesized. They are synthesized by the body. Non-essential means what? They are synthesized by the body. Now, what do you think can make something that was synthesized in the body not to be present in the body? It is when one or more of the organs synthesizing those molecules has issues or health issues. So when an individual is suffering from certain health issues, some of the amino acids that are synthesized by the body will not longer be able to be synthesized, and hence such amino acid has to be taken in our food in order to balance up the deficiency. So such amino acids that were initially non-essential but becomes essential due to the inability of the body to synthesize them are said to be what? The conditionally essential amino acid. They are said to be what? The conditionally essential amino acid. And two of the conditionally essential amino acid we have histidine, tyrosine, and Genic. All these ones, they are conditionally essential. And Genic, cysteine, glutamine, proline. Let me take example: tyrosine, and Genic, even histidine. We have another example: glycine, cysteine. Glycine and cysteine. All these are conditionally essential amino acids. Are we getting it now? So that is based on the nutritional requirement. Based on nutritional requirement, we classify them into the essential or indispensable. Essential or indispensable. We have the partial essential or semi-essential. Those who are required at infant age, required at infants, but are not required when you become what? An adult. Those ones are semi-essential. That is, they are required at one stage of your life, but as time progresses, they are no longer what? Required, because by then your body must have matured. All the organs that are responsible for synthesizing them must have started carrying out their various functions. Those ones are said to be partially essential or semi-essential. Then we have the non-essential. Those ones that your body can synthesize, hence you don't need to take them into your body for food or as food for growth and metabolism. Are we getting it now? Then we have the conditionally essential amino acid. The conditionally essential amino acid. Those ones that are essential or essential as a result of the inability of the body to synthesize them at a particular period in time. Now, that is classification based on the nutritional requirements. So let us go to the numbering of the amino acid using arginine. Now let us use lysine to number the amino acid. If this COO minus, then we have C, and here is NH3 positive. Here is what? H. Then we have CH2, 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 and then we have NH2. Or when it becomes ionized, it becomes NH3 positive. Now, if this is lysine as an amino acid, how do we number them? not using the 1, 2, 3 notation. Let us use they use the Greek notation, to number the amino acid. Like the way we have A to Z in our English alphabet, the Greek have alpha to omega in the Greek alphabet. And we are going to use the Greek notation to number them. Now, we said that this carbon here directly attached to the carbon dioxide and the amine group is called the alpha carbon. That is why we said all amino acids are alpha amino acid. Then after the alpha carbon, the next carbon is the beta. Then we have the gamma. Then after gamma, we have delta, which is your partial negative charge that we used to do. Then we have zeta, we have eta, we have theta, we have iota, we have kappa, we have lambda, we have mu, etc. Are we getting it now? So all those ones, they are the Greek alphabet. Are we getting it now? Let me rewrite them here. Alpha, beta, gamma. Gamma. Then we have delta. We have the delta, which is partially negative. We have the epsilon, which is represented with E. Then we have the zeta, which is this symbol here, something like this. And then we have the eta, the theta, etc. So any amino acid, no matter how long it is, follows this precept. We have the alpha. Then after the alpha carbon, we have the beta carbon. After beta carbon, we have the gamma. Then we have delta, etc. So that is how to number the amino acids. So this is the correct structure. Now, after the gamma, what are we going to have here? We have after delta, we have the epsilon, which is represented with E. Epsilon represented with E. So this is how to number the amino acid. So if you are given any amino acid that you enhance to actually number it, you follow this precept, which is the Greek notation. So that is that for the numbering. Are we getting it now? Now the next is the properties of amino acids. Now all amino acids are soluble in water. They are soluble in what? In water. That is one property. And then another property is they are colorless. They are colorless and then tasteless. All amino acids are colorless and tasteless. So those are the properties of amino acid. They are colorless and tasteless. Now glycine, alanine, tryptophan, and then histidine are sweet in taste. They are what? Sweet. Then leucine is tasteless. And then isoleucine and arginine are bitter. Isoleucine and arginine are bitter. So these are the physical properties of the amino acid. Are we getting it now? They are all soluble in water. They are colorless and some of them are what? Tasteless. But glycine, alanine, tryptophan, histidine, they are sweet. Leucine are tasteless. Isoleucine and arginine are what? Bitter. Are we getting it now? So we cannot classify them as generally tasteless. Let's just say they are colorless generally. Okay, that is the properties. Now the next thing is, this is where we are going to end. This is where we are going to end for this lecture. In our next lecture under amino acid, we talked about the ampholyte and the isoelectric pH. Thank you for your time.